So thank you for the opportunity to share my work on tunable hydrogel scaffolds that can be used to support neuronal networks in 3D. Every biological system and organ has a specific structure that gives rise to a particular function. And if we want to understand processes from development to disease progression, we really need to understand this particular relationship. To do this, we have two paths. We can take a top-down approach. So here we start with the full organ, in this case the brain or brain slices. And then we have implantable devices that we use to understand these systems. In the case of the brain, however, the devices that we have are quite rigid and they tend to cause quite a bit of compression and damage to the actual tissue. And at the same time, we're only able to record from a fraction of the cells at a time. On the other side, we have a bottom-up approach. So here we put a specific number of cells of a specific composition, and we can place them either in really defined scaffolds or in precise neural networks. And the advantage here is that we have control over the entire system, so if we wanted to change the scaffold mechanical properties or screen a drug through our precise networks, we're really able to do this. Of course, the disadvantage is that these are artificial networks. These paths actually meet again when we think about the existing material limitations in these systems. So despite the very different directions, the materials are actually very similar. We tend to use metals which are very conductive but also very rigid to record signals from cells. And these are also materials that deform plastically, so it's permanent deformation. We then have plastic layers as encapsulation, which have a very similar mechanical and materials profile to the metals that we're using. And there's been more work recently using elastomeric materials, which are a bit softer, but they're still, as you can see here, more than 1,000 times more stiff than neural tissue, such as the brain. And then one word that you can see a lot for all the living tissues is that every living tissue from brain to bone is viscoelastic. So it has a time-dependent behavior to applied forces, and none of the materials in our existing toolbox are similarly viscoelastic. The second limitation is more on the local scale. So if we really zoom in on this electrode tissue interface, we see that the structure of the metal is quite continuous versus the brain extracellular matrix below is porous. And then it's viscoelastic and porous, which allows cells to pull on the matrix and migrate through, which they're not able to do with these gold electrodes, and this limits our resolution over time. The hypothesis of a class of materials that can overcome both these limitations are hydrogels. So hydrogels are polymer networks that are swollen with water. And as you see from this range, you can actually tune the mechanical properties so that the modulus matches many biological tissues. Hydrogels can also be quite viscoelastic, and they're hydrophilic, similar to our tissues. So today I'll describe some hydrogels and methods to tune their mechanical and electrical properties, show that these can be used to then integrate neural cells, and then finally talk about integrating hydrogels to make precise 3D networks. There are a lot of different hydrogels out there, and we have a lot of parameters that we can consider in choosing our composition. So mechanical modulus and viscoelasticity we've talked about. Ligand density is important because this is actually where the cell is attaching. So in the case of natural components like collagen, it's already there. But for synthetic materials, we tend to incorporate RGD or other ligands for cells to attach to. And then the mesh size of the material is important too, so that components from the media can diffuse through to the cells. And then we also have a lot of tunability in the type of crosslinker that we're using. During my PhD, and for the first two parts of this talk, I'll focus on alginate. Alginate is really advantageous because you can independently tune all 5 of these mechanical properties. So you can pick a viscoelasticity, and without changing that viscoelasticity, you can change the mechanical modulus. And it's also advantageous because it's already approved by the FDA for bandage dressings and even food stabilizing agents. When we think about the properties that we're looking at the G prime, so how elastic a material is, the G double prime, which tells us how viscous a material is, and then the ratio of these two, G double prime to G prime, gives us a sense of viscoelasticity. So the very first step of my PhD was really characterizing my tissues of interest, so in this case the heart and the brain, and then seeing as I change the amount of crosslinker for the alginate, how we can match both the G prime and G double prime of these very different biological tissues. So right now, the alginate is ionically conductive. So when we think about properties of metal to record from cells, the alginate would do a pretty poor job at this. But we can incorporate electrical conductivity and functionality into these materials by starting with our alginate matrix and then mechanically embedding different nanomaterials, so metal nanoparticles or carbon nanomaterials. And again, 2 flavors for this. We can start with our matrix of alginate, mix whatever components we want, and then cross-link the gel. And here we get a nanoporous formulation, which is the same mesh size as alginate on its own. But we heard a very beautiful opening talk about freeze drying. So we can do the same thing with our system, where we mix our components, but before cross-linking, we freeze the gels, we dry them, and then we cross-link the system. And this, for us as well, introduces a microporous architecture. If we look at this under SEM, we can see on the left the nanoporous alginate, and again, with no additives, we now have a microporous formulation with quite large pores. And this is actually really advantageous when we start making electrodes, because here I have an alginate schematic. Alginate is the blue rectangle, and then in this case, I have 67 additives, which are the black circles. And if we randomly mix and cross-link our structure, I've drawn the percolating or electrical path here in red. You can see it's discontinuous. But for the same geometry and number of particles, if we instead have ice crystals and freeze the gel, we then displace all the conductive particles around the ice. And then when we remove the ice through lyophilization, we have this porous structure which we can cross-link and preserve. So then with the same number of particles, we're now able to have that percolating path. And it's important because it's really the alginate that is so soft and viscoelastic. So the more of these additives that we're adding, we lose that viscoelasticity. So we really want as few additives in our system as possible. Going back to the SEM images, so for most of my PhD, I worked with graphene flakes, which is the second column. And these are an aqueous dispersion. So you can see that the flakes are able to uniformly coat the wall of the scaffold. You can also look at carbon nanotubes, which is the third column. These are hydrophobic, so they tend to form dense bundles together. But then in the last column, you see a combination of graphene with carbon nanotubes. So here you see the coating along the wall that bridges these dense bundles together. And if we now look at the function of these electrical scaffolds, we can see in red the microporous formulations. So as expected, all the microporous formulations are more conductive than the blue nanoporous formulations of the same composition. And then again, we can see why we want a combination of graphene with carbon nanotubes, because for a lower total amount of additive, we have formulations that are more conductive than the carbon nanotube equivalent, even with more carbon nanotubes added. So then the last part of this work was starting to integrate cells. First, we looked at rat primary cells, placing them on top of these scaffolds. And we wanted to compare viscoelastic and elastic substrates that were either soft, so 1 kilopascal, or stiff at 6 kilopascal. And the neurons here are shown in green with a beta-3 tubulin stain. And you can see that on the soft viscoelastic case, we have the highest coverage than any of the other cases. And this is really the properties that match that of the brain. What was also interesting is that the next best coverage was the viscoelastic stiff. So these primary cells really preferred viscoelasticity over the stiffness of the substrate. And then for both elastic cases, you can see that they were quite similar. So this transitions to the second part where we really wanted to investigate how we can use these scaffolds to grow different types of cells. If you put alginate under SEM now, you can see that it really matches the structure of extracellular matrix that is normally found in the brain. It has a similar porosity, and it would allow cells to migrate and remodel. So here we were really looking to make our mechanical properties as similar to fresh brain as possible. Now we were using nanoindentation, so our target mechanical property value is 5 kilopascal. And we can change, in blue is a 1% concentration solution of alginate, or 2%. And then we can span an entire range of carbon content and see that we have between 0 and about 0.4% carbon in our gels while still matching our target mechanical properties. On the right is the tan delta. So you can see the viscoelasticity of fresh lamb brain in green None of our formulations are quite as viscoelastic, but again, if we compare to the existing material toolbox, we see that we're much closer to that of the brain. In this work, we were also looking at electrical stimulation, applying exogenous electrical stimulation throughout the scaffold. So for this, we wanted a minimum conductivity of a few Siemens per meter. And then this gives us the upper bound. So we can see that with more than 0.1% carbon in our compositions, we can have gels that have this minimum conductivity that we need. And then this gives us a range between 0.1 and 0.4% carbon in our scaffolds. And then the last ingredient is really the cell type. So we started with primary rat neurons. We also used NGN2-derived neurons. But then ultimately, most of this work was done with NPCs, so neural progenitor cells derived from iPSC. And NPCs are really advantageous because they naturally form neurons, astrocytes, and oligodendrocytes over time. So the metrics we were looking for a healthy culture, we wanted to see if we had a high percent of our stem cells differentiated after a 5 or 6 week time point. We were looking for dense neurite networks, and then we really wanted to see if we could get all 3 cell types present. For this experiment, we kept the cells in culture for 43 days. We kept them inside the scaffolds for as long as 12 weeks. But then at the 43-day mark for this batch of experiments, the neurons are shown in green. I see that my hair is blocking a lot of the markers at the bottom. But neurons are in green, cis beta-3 tubulin. And again, we're comparing viscoelastic and elastic substrates, and the top column is the amount of carbon that we're adding inside the gel. So we can see, if we quantify, that in our cases of viscoelastic substrates, we have a higher percentage of neurons that have differentiated inside the scaffolds. And then also, interestingly, as we increase our scaffold conductivity, we see even more neurons in the system. At the high conductivity as well, you can see that the cells are really forming networks around the pores rather than a continuous sheet or aggregation. The next marker were astrocytes. So for the astrocytes, we use GFAP. GFAP shows inflamed or activated astrocytes, and you can also identify that these are activated cells by the morphology of the cell. In the bottom row for the elastic case, especially for the high conductive gels, you can see very, very big diffuse cells. And this makes sense because an elastic substrate mimics more of a diseased brain rather than the healthy state. Versus in our viscoelastic substrates, the astrocytes have a really small phenotype, and you see really long extensions that form, if you look in the Z stack, underneath the neurites. So they're really having this supportive role. And then additionally, if we quantify the percent of astrocytes in our culture, we see that the viscoelastic substrates have more differentiated astrocytes, which again increase with scaffold conductivity. And then the last marker we were looking at for this batch of experiments were oligodendrocytes. So here we looked at myelin basic protein, which is shown in pink. And we were really excited to see that in the viscoelastic substrates, especially the conductive scaffolds, we have quite long stretches of myelin, and there's much more myelin present in our viscoelastic substrates than the elastic substrates. And again, as scaffold conductivity increases, we see more of this myelin present. I'll just make a small note. This is not just a system for neurons. Fibroblasts are also responsive to electrical stimulation, so then we had another study where we applied electrical stimulation in the Z direction, and you can see too that you're able to now migrate and direct the migration of fibroblasts towards the bottom part of the gel if you apply the stimulation this way. So it's an exogenous stimulation that is applied throughout the scaffold. To quickly conclude this part, we have scaffolds where we can tune independently mechanical, electrical, and then the cellular components, and use these scaffolds to form really beautiful neuronal networks in 3D. So in the last part of my talk, I'll focus on what I'm working on now as a postdoc. So the lab that I'm doing my postdoc in recently described these PDMS microstructures, and you can pattern and design the PDMS in a way that you have seeding wells, which are open from top to bottom, and you can either put cells or spheroids inside. And then you have these small channels underneath, which are just thin enough to let an axon grow through. And this platform is also compatible to be put on top of a multi-electrode array, so you can record from cells throughout the studies. The channels on the microstructures underneath don't have to be simple lines. You can pattern them in a certain way so that you have directionality in your system. Here, both cell types are the same. They're just colored differently, and you can see that with this patterning, you're always growing red to green rather than having the green grow backwards. There are some cases, though, since all of this is in 2D, where you might want a 3D system to mimic physiological processes. And in these cases, and my goal as a postdoc, is to integrate hydrogel inside these systems. So the idea is you have the same setup, but hydrogel everywhere, and then you can seed your spheroid, which is again on top of a multi-electrode array, and then image, but also record as the spheroid is growing over time. To see in our system if 2D or 3D has a difference, we're using sensory neurons, which are iPSC derived, and on the left is SEM image of the cells that are grown following manufacturer instructions, so in 2D, and then on the right is our 3D encapsulated cells. And you can see that the cells on the left are very large, they're quite flat. So if we think more from the mechanobiological perspective, the forces that these cells' nuclei are sensing are very, very different than the round cell on the right. But in this case, the big question is how do they look in vivo? So from this study in 1996, they took the chick DRG neurons from an embryo, and you can see that the cells in vivo really do have this round phenotype. So in the goal of a platform that's a little bit more physiologically mimicked, the hydrogel is helpful for that. We can fill our structures with photocross-linkable hydrogel. So here we're using GelMA. And just as in part 2, where we can change and optimize a whole range of properties, we can do the same again. I'll skip this slide briefly in just looking at different compositions and show that for our GelMA, we can support our spheroids actually for more than 100 days. But in this image, the spheroid is at 60 days. The green components are shown. The green components show the alive portions. So you can see everything is alive, and the gel is 500 micron thick, and the spheroid is really able to form neurites throughout this structure. And then we're also able to compare 2D and 3D, the same kind of microstructure, and we're able to see some differences as well. So if we look at 25 days for both systems, this is using the same batch of cells, you can really see in 3D that you have a fasciculation forming, so the neurites are converging and forming a nerve bundle. So for applications where we were interested maybe in more nerve on chip, it's really nice to have this 3D platform, and we have these nerves bundles forming. We have started to interface our platform on top of a high-density CMOS multi-electrode array. This is from Maxwell Biosystems, a startup out of ETH. And we can see really preliminary recordings that in this tiny red channel, or channel highlighted in red, we're starting to be able to record some action potentials in our system. So I just want to give a few broad conclusions. Hydrogels really mimic the mechanical properties of neural tissues, and they can enable cells to have more physiological responses. And then we can also create viscoelastic electronics that can be used as scaffolds for in vitro platforms and apply exogenous electrical simulation throughout. I wanted to thank my PhD lab, so it's the lab of Dave Mooney at Harvard, as well as my postdoc lab, Janos Voras at ETH, and give a special mention to Blondine, who's a very talented PhD student who's closely working with me on this last project. And then before questions, LBB had quite a few posters, so I just wanted to highlight some of them. Unfortunately, all in the past, but I include the abstract ID so you can look them up, and most of them are sitting here. But everything ranging from different micro or even nanostructures to more automated processes, such as placing spheroids inside our microchannels. And thank you all for your attention.
Thank you. Any questions from the audience? No? Wait.
Thank you. Really, really lovely presentation. On the last bit, when you showed the 2D versus 3D, I don't know if it was just about the focal plane, but there were these sort of blobs on the 2D neurons that weren't there in 3D. Is that just an artifact of 2D, or what is?
So you're referring to, sorry?
All the punctate spots along the 2D
Yeah, I think this is just an artifact. We see over time that they're not cell bodies, but they're these round features, both in 2D and 3D, that can form. In the terms of focal plane, though, you don't see the spheroid in 3D because this is a focal plane that's in focus for this nerve structure.
Any more questions?
Sorry for making you running. Thank you for the presentation. Hugo Salmon, Université Paris-Cité. Can you hear me well?
Yeah?
Yeah. Regarding the better myelination you observe in your tuned viscoelastic versus elastic structures, is there a possibility that the optical performances of your hydrogel is better in viscoelastic than elastic, and that you observe more myelination because it's more transparent? I'm not familiar with viscoelastic versus elastic optical.
Yeah, it's a good question. So the difference between a viscoelastic and elastic gel, you start from the same starting material, which is the elastic one, and then the difference is the molecular weight of the polymer. And we change it by just irradiating the starting material. So the optical properties are almost the same, especially when we start to add these additives. It's really just the mechanical properties that are changing.
Maybe one more question.
Hi, thank you for your talk. And I was wondering if you tested the mechanical properties of your gel, but over time, if you see any decrease in rheological capacities and so on. Thank you.
Yep, it's a great question. So once we make the gels, we actually soak them for 30 minutes. They swell a little bit, but after this 30-minute window, they really plateau off. So the mechanical properties don't change. We've tested them 30 minutes after, or an hour after formulation, 4 days later, 7 days later, and there's really no significant change in the mechanical properties.
All right. With that, thank you, Christina.
Thanks.